everyone. In this video we will learn how to design the primers for our PCRs. Primers can be designed for the genes, or if we don't know any genes, we can do it for the mRNA sequences. That is, in database we have to select any genes if we know their names, or otherwise if we can also take that protein's name. That is, for example, I will take here MMP7 protein, that is matrix metallopeptidase 7 from the Homo sapiens. So here we get that the gene that is from the Homo sapiens, it is also known as MMP7, MPSL1 or PUMP1. It is PUMP1. So gene ID for this protein is 4316. Here, to design the primers, we have to go for reference sequences. Here we have the transcript and its ID, accession ID we call it as. So it has 267 amino acids, the proteins. We have to click on the transcript accession ID and it will directly go to the GenBank format of that reference sequence. Here we get the various informations about this sequence. So after that we have to go on the right side, that is analyze this sequence. Here we have to click for pick primers. As I move the cursor to the pick primers, here we can get design and test primers for this sequence using primer blast. Here we are using primer blast, that is a database of using NCBI database. Here if we know the accession ID, we can directly come to the primer blast page and we can add the accession ID, or we can also add the FASTA sequence, or we can also upload the FASTA file. That is, these are the primer parameters: PCR product size, that is primer melting temperature, exon junction spam, exon junction match, intron inclusion. Next, the primer pair specificity checking parameters, and after that here last there is a get primers. We have to click on that get primers. After clicking the get primers, the pages will go to the next one. Here we will get the page of the primer blast where we can get the status, submitted, current time. We can also check the status of the designing of the primers. So it will take some times for making primers specific to our PCR template. So when the primers are ready, we will get this type of the results, that is primer blast results. Here the first one will be the input PCR template, that is the mRNA reference sequence which we have selected, and here is the specificity of the primers, that is organism limited to Homo sapiens only. So here this will be the graphical representation, graphical view of primer pairs, that is the primers which we have designed. So here detailed primer reports. Here we get the primer number 1, primer number 2. So like this I have designed 8, 9, 10, total 10 primers are designed for this mRNA reference sequence. So after this one we can go here, that is the download primer pairs. In this we can get text format, we can download it in the text format, and we can also download it in the tabular format. So for here reference, I will download it in the text format. I also download it in the tabular format, which can be saved for our future references. That is, if I can open that one, so primer 1, that is, it is in the text format. We get save results like this one. It is in the text format, and if I go to the, it is also in the text format only, but this will come in the Excel sheet. Like this we get in the Excel sheet. It is not actually the Excel sheet format because I don't have this Excel software in this one, so this will comes in the Excel sheet format. So this concludes the, we can also, that is the CSV format. In CSV format also we can download it, but it is I can't open because it doesn't have any related softwares which can open the CSV type of files. So this completes the video on designing the primers